Good morning, everyone. Thank you for joining us today. Can you hear me well? Okay, perfect. Thanks for joining us today. So today what we would like to do with my colleagues is to walk you through one of the models that we're using, which is a model which exhibits a human immune system, and try to go into detail on what do we know about that human immune system, which are its features, how functional it is, and how could this be applied to target validation, to efficacy assessment, and then at the end we'll talk about safety assessment of therapeutics. Okay? So my name is Kader THIAM. Again, thanks for being here with us today. So basically, as you know, so just a disclosure, I work for genOway's, obviously, okay, and I'm also a stakeholder of the company. So as you know, one of the main challenges in the field when it comes to a model with a human immune system is to have a model which has both lymphoid and myeloid cells. Humanized model has been developed for years and years and years. Most of them have a good functional lymphoid compartment. However, when it comes to having a model which has both lymphoid and myeloid cells, this is where it's becoming more and more challenging. The reason why this is key is that most of the therapeutics which are being developed at the moment play on both axes, try to activate CTL response while also inhibiting the suppressive activity of some of the myeloid cells, or use a myeloid cell to trigger functional activity. This is basically the reason why it is challenging. These are the models which are the most known in the field and the most published. The NSG-SGM3, that model has been developed several years ago. It overexpresses 3 human cytokines. And those human cytokines which are overexpressed, namely SCSF, IL-3, GM-CSF, will indeed drive the development of a human myeloid cells together with the human lymphoid cells. Myeloid cells will be present, but the main limitation of the model is the lifespan. The lifespan of the model is rather short, around 4 months, and the higher is the chimerism rate. So the more human cells you have in your model, the shorter is the lifespan of these animals. The other model, which is also well known in the field, is the NOG-EXL. That model overexpresses 2 cytokines, GM-CSF and IL-3. They also drive a myeloid compartment, which is present. Those animals have a longer lifespan. However, again, the higher is the chimerism, the sicker the animal gets. So basically, having both systems co-developed in these mice creates a phenotype. When we look into this, one of our explanations was probably coming through how these cytokines were overexpressed. Our cytokines are overexpressed here using ubiquitous promoter, meaning that when the human myeloid cell will develop and will get activated, their cytokine production will never downregulate these cytokines because these are synthetic promoters that will be totally insensitive to any regulation by human cytokines. So we decided indeed to work on a different approach where we will have both lymphoid and myeloid cells have a fully functional myeloid compartment and lymphoid compartment while avoiding having side effects due to a myeloid cells development in vivo. How did we achieve this? For that, we decided to take advantage of a model that was developed by James DeCento at the Pasteur Institute in Paris. This model is on a biopsy background. Those mice have no T cell, no B cell, no NK, like the others. The main difference is here. These animals have a knockout of a gene called FLT2, also known as FLK2, and that FLT3 knockout results into reduced mouse myeloid compartment. That reduced mouse myeloid compartment frees up a niche for the human cell to develop upon reconstitution with cord blood cells. So when the mice are reconstituted with cord blood cells, they will develop a human immune system. That human immune system will first be strongly dominated by lymphoid cells, but upon FLT3 ligand injection, FLT3 ligand will selectively activate the human cells and won't be able to cross-react with the remaining mouse cells due to the FLT3 knockout that has been performed here. And the human myeloid compartment will start to develop and to become activated by the stimulus. So how this system differs to the existing models? First, we are not on a SCID background. We're on a RAG2 background. Why do I emphasize this? SCID, it's a mutation in a gene called PRKDC. PRKDC is involved into DNA repair mechanism, so those animals develop spontaneous lymphoma as they aged. We are on a RAG2 mutation, so we don't have that drawback. Since we don't have a SCID mutation, mice could be irradiated because the DNA repair mechanism pathway is not affected. So you can basically apply standard of care to those mice. And the complement cascade is functional, which is not the case for any mice deriving from the NOTS background, because NOT mice are C5a deficient. Right, now let's focus a little bit on how the immune cells look like into this model. Upon reconstitution of those mice, if we look into the different organs, this is the type of human cells that we found. This study was conducted on mice coming from 5 different donors, and the animal were between 18 to 22 weeks old. And as you can see here, looking into bone marrow, thymus, liver, spleen, blood, and lung, we clearly identified that the colonization of these tissues is dependent on the tissue we're looking at, and the colonization is also consistent with the biology of that tissue. So for example, in thymus, you'll see mainly T cell. In the liver, you'll see NK cell, monocytes, B cell, T cell, and monocytes. And in the lung, you'll see the same type of composition. What is quite important into this setting, as you could see, we're getting NK cell. They are present. There is no IL-15 added, no IL-2 added. I'll get back to this later. If we look now about the engraftment over time, in the study that I'm showing here, the authors have evaluated the colonization of blood, spleen, bone marrow over 51 weeks. So basically, the kinetics is here, blood, spleen, bone marrow, blood, spleen, bone marrow. And as you can see, that remains stable with time. So there is no obvious GVHDs that develop in those mice, and you can age them up to a year. We never went further, but after a year, nothing strange was developed in the animals. How are the immune cells now that develop? Looking into the B cell compartment first, as you could see, we see a different subset of immune cells which are known for B cells. And when those B cells are recovered from the mice and activated ex vivo with an anti-CD40 and a cocktail of cytokines, they shift toward IgG negative cells or IgG negative IgM plus cells to cells which are IgG plus IgM plus, so they're becoming indeed mature B cells and activated B cells. Looking into the ability of these B cells to produce Igs, we conducted a study with one of our partners, which wanted to know whether those B cells will be able to secrete IgE upon activation. So we were not in an allergic setting, just a general activation, and see what are they able to secrete. And as you can see, when LPS is injected at 2 mg per ml into the animals, we detect 24 hours later a IgE production. But that IgE, as you could see here, is donor dependent. Okay, so here one donor produces IgE, while 2 other donors did not. When we look into the class switch into this animal, we were able to see class switch up to IgA. So the whole class switch is able to happen. Also, I should say that the IgE titers in the model are low compared to what you have in mouse, but also compared to what you have in human. Now, what about the T cell subsets? Looking into the T cell subsets, as you could see, if we look into the spleen of these mice, we'll basically characterize some TCR, some T cells which are CD4 positive, some CD8 positive cells, some Tregs, but also some gamma delta T cells. And that's quite unusual for his mice because his mice generally have very few to no gamma delta T cell. I'll get back to this in a few minutes. Looking now into the T cell subsets per se, we basically characterize the standard central memory, effector memory, EM array, and naive T cells. Let's focus now on the gamma delta T cell because I just said that that was a surprise for us and for others because his mice are known to have little to no gamma delta. Looking into the gamma delta T cell, here we look into 4 different donors. We basically detected gamma delta T cell in blood, spleen, bone marrow, both in frequency and cell counts. And here we compared it to a healthy human donor that we use as a positive control. When looking into the subset of those gamma delta T cells, then, these gamma delta T cells were basically in the blood dominated by delta 1 and delta 2, which indeed doesn't really reflect the human situation because in human situation, you have approximately the same frequency of delta 1 and delta 2 and very few delta 3. Here we had much more delta 1 than delta 2. And when looking into the tissues, we found that we had delta 1 and delta 3 that were dominated the gamma delta compartment as reported in the literature. So, are those cells functional? And then we get into tumor-bearing mice in a few minutes. First study that we did was just to run a very basic vaccination study in the animal. So mice were received FLT3 in order to boost the myeloid compartment, have the APC being present, and then vaccinated with KLH. And then they received 2 boosts, and after the last boost, 2 weeks later, mice were taken down and the splenocytes were analyzed. First studies that we run was an ELISpot to see whether those T cells could get activated in an antigen-specific manner. And as you can see, upon restimulation with KLH, we clearly see a gamma secretion from those T cells. So we have a T cell activation which is antigen-dependent. This indeed corroborated a study that was run a few years ago by another collaborator of us, Bernard Meyer at the CEA in Paris, where this guy did the same study and added an anti-HLA-A2 in order to show that the antigen presentation will be dependent on human APC or mouse APC. And that you can see, adding an anti-HLA-A2 shut down the antigen-specific T cell response. In his study, he was able also to see IgE titers as, antigen-specific IgEs, looking into IgM and IgG. We are trying to reproduce this. This has to be confirmed so far when it comes to the antigen-specific Ig response. Now, what about the gamma delta? We said they were present. Are they functional? So we decided to use a compound developed by one of our partners. That compound is a delta-1 targeting agent. This partner was used to use this compound with some human PBMC, and he established that this compound, when incubated with human delta-1 positive cells, triggers an activation of those T cells, which could be seen through an increase of CD69, CD25, and at a lesser extent for 1BB. That was human PBMC from a healthy donor. What happened if we reproduce the same study with cells coming from the mice? As you could see here, using cells coming from the animals, we have the same activation pattern that is reproduced. So the T cells, the gamma delta T cells, the delta 1 T cells that develop in these mice respond similarly to cells coming from healthy human donor. We repeated the same study, this time using a gamma-9 expander. Gamma-9 is common to delta 1 and delta 2 T cells. And when using the gamma-9 expander, we also observed a significant increase of delta 2 gamma-9 cells in terms of frequency. Same thing was also observed for delta 1 gamma-9. And when looking into those cells, they show an increased CD25 expression, so increased T cell proliferation markers, and also cytokine secretion through gamma and perforin. So T cells are present, they could get activated. So far, the main difference with the other model is the gamma delta T cell. What about the myeloid cells now? This is, before I move to myeloid cells, let's talk about how this T cell would behave in the context of tumor-bearing mice. So a study was conducted by Capasso and colleagues in the lab of, I forget her name. Doesn't matter. In University of Denver, Colorado. And mice were inoculated with a triple-negative breast cancer cell line and treated with nivolumab. TILs were analyzed at day 11 and day 21. And basically, this is what was reported in this paper. Nivolumab treatment resulted into a tumor growth control. Looking into the TME of the animals, there was no significant changes at day 11. But at day 21, they observed an increase of CD8 T cells. And these CD8 T cells were cytotoxic T cells as being interferon gamma positive and perforin positive. Looking into the TME at day 21, they also reported a decrease of NDSCs. And model was used in order to test some combination therapy between PD-1 and HDAC inhibitor, but also PD-1 and MAP kinase inhibitors. So the T cells are present, they get recruited at the site of tumor development, and they could get triggered and activated by known compounds in the markets. Now, let's move to the myeloid compartment. How are the myeloid cells? The myeloid compartment will be triggered upon FLT3 ligand injection. So it's a transient boosting. How does it work practically speaking? Mice are injected once every 2 days with FLT3 ligand. And at the end of the last injection session, you start to see a significant increase of myeloid cells in general, in blood, spleen, but also in bone marrow. And that could be seen looking into different subsets of myeloid cells, such as CDC and PDCs here, but also looking into subset of CDCs, such as CDC1, CDC2, and also looking into, for example, macrophages in the bone marrow, as shown here in the slides. This boosting is transient. So if you deal with naive mice, if you don't provide any other stimulus, those cells will go down 3 days later. But you have the possibility to reboost it when you wish to. When you will deal with mice that carry tumor, it's a different setting because then the tumor will activate those cells and they're going to be maintained. And I'll show you this in a few minutes. Before moving into this, let me just emphasize a little bit on what are the remaining mouse cells. Because, of course, the challenge in the HIS mice is how the murine response will contribute to the human response that you would monitor in your model. We said that at the beginning that the FLIC2 knockouts, also known aka FLIC3, result into reduced mouse myeloid compartment. What does it mean, practically speaking? We just run a study where we focus our flow analysis on mouse CD45 positive cells. And we compared the HIS mice to wild-type bulbous C. And what you could see here is that you have a drastic decrease of mouse myeloid cells. This doesn't apply to the neutrophil compartment because the neutrophil is not FLT3 dependent. And upon injection of FLT3 ligand, it doesn't activate the mouse compartment, whereas human cells get activated and proliferate, as said previously. Just to make it clear, although we see a significant decrease, in those models you have much more mouse cells than human cells. So count-wise, you will still see a significant amount of mouse cells that remain, okay? Just to avoid any misunderstanding here. Now, how the immune response behave when mice are treated with known therapeutics. The first study I'm going to share with you was a study which has been published a few years ago, where this group here developed an anti-PDC. So that their antibody targets CD303, and that antibody is indeed an IgG1 which has been modified to decrease defocalization, and that antibody has a high binding affinity to Fc gamma R3A, and it works mainly through a DCC, but also through a DCP. When this antibody was injected into the model, one injection at 30 milligrams per kg, this is what we observed. 1 day later, we start to see a depletion of the myeloid cells, of the PDCs in the blood of the animals. That was still visible at day 3. And at day 7, you have the renewal of the PDC compartment, which is visible here. Looking into the spleen, that depletion is shifted and start at day 3 and day 7. And in bone marrow, we haven't seen any depletion at all, so the antibody did not indeed reach the bone marrow and was not efficient in the bone marrow. We decided to repeat this with different types of therapeutics, and we use an NK engaging agents. I said earlier that we are not adding 15, we're not adding IL-2, and we have NK. Are these AAK capable of cytotoxic activity toward target cells? So we use an antibody which targets B cells. First question that we address is whether the NK cells in those animals could get activated, and could this work through the crosstalk between DCs and NK cells? So mice were injected with poly-IC, and as a readout of DC activation, we measured IL-12 secretion. As you could see, poly-IC increases IL-12 secretion. When we treated the mice with poly-IC, we also observed an increase into interferon gamma positive NK46 positive cells, so NK cells. And those NK cells were also CD107 positive, so they get activated, despite the lack of exogenous 15 and 2 as a cytokine. Now, are those NK cells capable of depletion? This is what we assess here. We injected into the animals the NK targeting agent, and we looked into the blood of the mice for the target cells, which is B cell here in that study. As you can see, the Fc competent agents clearly show a depletion into the B cell compartment, whereas the Fc silence version doesn't show any effect at all. Now, we have myeloid cells. The myeloid cells are present and could get activated. What happened in tumor-bearing mice? I said earlier that the induction of the myeloid compartment was transient in naive animals. Upon inoculation of tumor cell into these mice, the tumor cell, in fact, will do the job. And in order to show that, we did a study where mice received or not FLT3 ligand, were further inoculated with A549 tumor cells, so lung tumor cells, and we analyzed the TME of the animal at day 41 post-inoculation. And this is what we observed. The TME composition was indeed dependent of the pre-treatment with FLT3. When FLT3 is provided, we have more myeloid cells, more T cells, and less NK cells. Looking into the T cell, the majority of the T cells that were recruited was indeed CD8 T cells. And that was due to this pre-sensitization here, so the presence of myeloid cells that are present. We repeated the same study with our colleague from OncoDesign, where they used 3 different tumor types. So 2 colorectal cancer, adenocarcinoma, tumor cell line, and 1 breast cancer. And as you can see, again, your tumor composition is totally dependent on the tumor type. So the dynamic of interaction between tumor and immune system remains, and this is what's going to drive how your TME will be in terms of cell composition. How now the myeloid cells in the TME evolve with time? So we took here as a model, model system the MDA MB231 cells, which are triple-negative breast cancer cell line, and we first investigated the TME when tumor reaches a size of 1,000 cubic millimeter. First, take-home message from this experiment. We enrolled 5 different donors. As you can see, the tumor growth is not donor-dependent. It's quite reproducible among donors. Now, what do we see in the TME when the tumor are around 1,000 cubic millimeter? The majority of the cells are T cells. We do detect some cells, and we do detect as well some NK cells. These NK cells are CD56 pose, CD16 pose, but also CD16 pose and CD16 neg. And among the myeloid cells, at 1,000 cubic millimeter tumor, we have mainly M1, M2 transitional-like macrophages and few M2 macrophages. So please just keep these 2 numbers in mind, 40%, 20%. Now, we repeated the same study with MB231, but this time we looked into tumor that were around 400 cubic millimeter, so much smaller. And we wanted to see whether the recruitment will be different and the activation stages will be different. This is what we observed. We have 15% of immune cell in the TME. Among those 15%, 2/3 are human cell in reds, 1/3 are mouse CD45 positive cells. When focusing on the human CD45 positive cells, these are mainly cells that are myeloid cells. These myeloid cells are mainly CD14 positive cells. They are M2 macrophages, which express ILT2 and ILT4, so RB1 and RB2, and those macrophages are activated macrophages. So you clearly see that the activation stages evolve with the tumor burden, but also with the type of tumor as shown previously. And this model has been presented and posted by Macromix yesterday, where basically they use this system, not this tumor model, but this animal model, to test an anti-LILRB2, LILRB4, and they were able to achieve a tumor regression in the system. Now, how this depends on tumor. We reproduce the same study, but this time we've used another tumor model, which is HPAF2, a pancreatic tumor. Using HPAF2, we basically confirm what we've seen previously. The tumor growth is totally donor independent. But you clearly see that your tumor growth is much slower than previously. What about the TME composition now? Here, it's another story. We still have 50% of immune cells in the TME. But the majority of those immune cells are then mouse CD45 positive cells. Only 1/3 is human. And that third is mainly CD4 T cells, very few myeloid cells. So we have in that TME very few human cells which are recruited. Despite this low number, are those cells capable of effector mechanisms? So this HPAF2 model was used in order to assess some T-cell engager, because we had a lot of T-cell present, although frequency-wise, count-wise, they were low compared to the mouse cells. Mouse were treated with, integrated with HPAF2, and then treated with a bispecific targeting CD3-TAA, or CD3-TAA in combination with the CD28-TAA. So basically, would a signal 1 and 2 on T cells provide a better cytotoxic activity than a signal 1 alone? The CD3-TAA was used as a suboptimal dose in order not to get the total tumor regression. And as you could see here, by combining it with a by combining the CD3-TAA to CD28-TAA, we clearly observed a quite nice tumor growth inhibition, which were correlated by the survival of the animals. Looking into the blood of these animals at day 14, we observe an increase of human T cells, CD4 and CD8s, but also increase of human-activated neutrophils and myeloid cells. So although there is few cells in that system, the system is appropriate for T-cell engager assessment. How donor variability now influences this type of results? I just shared with you the data as the means that we observed. What happened if we break it down per donor now? When you break it down per donor, you clearly see that you have a donor effect in the response. You do have 2 donors that respond pretty well to the treatment, and 2 donors that are much less responder to the treatment. Why am I showing you this? Because quite often when people come and ask for his mice, their first question is, can I get 50 mice from the same donor? Yes, that's feasible. But does it really make sense to focus on a single donor? What are you going to get out of your study if you don't have a result? Is that donor dependent? Is that due to the therapeutics? Whereas by mixing your donor and keeping the same number of animals in your old study, you have a better idea of what your compound does, and also what is the donor variability to the response you're monitoring. So myeloids are present, they get to the TME, they contribute to the immune response. What about now assessing the immune-related adverse effect triggered by your compounds? Several years ago, AstraZeneca published this paper in which they wanted to see if HIS mice could be used to assess cytokine release syndrome in vivo. So in this paper, these guys injected OKT3 to the animal, and they clearly show that PBMC, and I say PBMC, reconstituted NSG mice, show a very nice release of inflammatory cytokines. In the paper, the authors said that the downside of the system is that using a PBMC reconstitution, more than 90% of your cell will then be T cells. They reproduced the same study using NSG reconstituted, CD25 reconstituted NSG mice. And they observed very little to no gamma and TNF-alpha. When we saw these results, we said, okay, the test article was OKT3. OKT3 is going to bind to T cells. What's going to happen is that those T cells are going to secrete gamma and TNF-alpha. These 2 cytokines should in turn activate the myeloid cells, which will secrete, if present, IL-6, IL-1RA, TNF, and gamma, which will reactivate the T cells. So question was whether having a myeloid compartment will change this type of reaction. And this is what we published this year here with our colleague from Crohn Bio in a paper published in Frontier Immunology. We basically reproduced exactly the same study. Mice were treated or not with FLT3 and injected with OKT3. And these are cord blood cell reconstitution this time. This is what we observed. OKT3 alone results into an induction of both lymphoid and myeloid-based cytokine. But when FLT3 is added prior to OKT3 induction, so when the myeloid component is present, we enhance the cytokine production. And we also enhance the myeloid cytokine production in the system. So the myeloid compartment will indeed significantly contribute to this response. And we first see TNF and IL-2 at 6 hours. And then comes some myeloid cytokines, which are also present. And finally, we see the whole contribution of the myeloid compartment by having 6, CXCL8, and also IL-10 being produced. Now, do those animals develop, what is the activation status of the immune cells? We look into the impact of the treatment on myeloid cells present in the animal. And as you could see, FLT3 by itself, in blue, has no effect on the activation status of the myeloid cells. OKT3 activates myeloid cell, not directly, but through gamma and TNF that is produced. And OKT3, with the pretreatment with FLT3, indeed enhances even further this activation. It has been reported in the literature that OKT3 depletes T cells. So we wanted to see if we were able to see this. We look into the T cell compartment of the OKT3 treated mice. And as you can see here, we have a quite nice depletion of the T cell compartment when OKT3 is injected. We wanted to know if this applied to other cell types. So we checked for the monocyte compartment. And indeed, OKT3 also depletes monocyte cells. And that depletion is most likely due to an exhaustion because of the activation that I showed previously. Do we have any clinical sign in the system? When OKT3 is injected without FLT3 pretreatment, we see a body weight loss in the OKT3 treated mice, but no effects on body temperature drop. When myeloid cells are present, both clinical signs could be seen: body weight loss, body temperature drop. So the standard clinical signs of CRS are reproduced. What about mouse SAA, which is a marker of inflammation? Mouse SAA goes up, and that goes up together with the presence of the myeloid compartment. So the myeloid compartment enhances the cytokine release syndrome, but also contribute to the clinical sign that we are monitoring. Could we rescue the CRS using a TNF blocker? Infliximab blocks selectively human TNF-alpha. So what are we seeing here is that due to the human cytokine or the mouse cytokine that could be produced by the remaining mouse cells, we block TNF using infliximab. And as you can see here in orange, that totally mitigates the cytokine production, but also that mitigates the clinical sign as body weight loss and body temperature drop. So what we're monitoring, it's really through the human cytokine which are produced rather than the mouse cytokine, which could also be produced on top of the human cytokines. Then we decided to take compound that went into clinic and see how they activate the immune system in the system. So we took a compound called Blincyto, which target both CD3 and CD19. It's a bispecific. This bispecific, when used into the clinic, resulted into cytokine release, and we used exactly the same protocol than previously. And as you could see here, the injection of Blincyto into the animals result into a cytokine release. The cytokine release that we're seeing is no longer the same than previously in terms of kinetics, because the cytokine peaks at 2 hours versus 6 previously, but also in terms of cytokine which are produced. And again, one more time in this system, we were able to show that infliximab block the activity. Second compound that went into clinic which was tested was an anti-VISTA developed by Janssen, which has an early stop a few years ago in clinical trial due to some cytokine release that was observed. Just for you to keep in mind that VISTA, it's mainly expressed on monocytes. And when we check for the monocyte in the HIS mice, VISTA is strongly expressed, on the myeloid compartment. And when we check in the myeloid compartment, VISTA is strongly expressed on monocytes, but also on dendritic cells. So we tested the Janssen compound, which has an early stop in the clinic, and this is what we observed. When we compared it to OKT3, which is our positive control, we had a quite drastic gamma and I1 array production compared to OKT3, which indeed was in line with what they reported. In the paper that they published in 2020, they only reported cytokine release. So we went further. We tried to understand what could be the cause of that cytokine release. We investigated different immune cell population in the anti-VISTA treated mice, and what we observed, indeed, is that looking into the VISTA-expressing cells, so the monocyte, we see a very drastic depletion of that compartment upon treatment with the anti-VISTA. So we said, okay, could we now use this to test a new generation of anti-VISTA? Our colleague from Sensei Biotherapeutics developed a new anti-VISTA which works in a pH-dependent manner. That antibody would bind to this, will block the binding of VISTA to PSGL1 in an acidic milieu in the tumor nests. So that compound works only in acidic condition. When we tested this new anti-VISTA, we had indeed no cytokine release at all compared to the previous version. And when we study the different immune cell population, we clearly show that that anti-VISTA that doesn't trigger cytokine release doesn't deplete neither the myeloid cells, in the model. So today, the model is being used a lot for this type of study as well, but doesn't mean that only his mice should be used to assess therapeutics. In fact, the model becomes complementary to existing model. The VISTA study that I just showed here has been published 2 days ago in Nature Communication. And as you can see here, the model, the effectiveness of the compound, the MOA, was investigated into knock-in mice, while its safety profile was investigated in the BRGSFis mice. And the nice part of the story is that when Sensei Therapeutics bring this compound to clinical trial after the BRGSF study, they first established the PK profile of their compound, and then they did repeating doses of their compound into patient, and looking into gamma 8, 10, 8, and TNF-alpha, no cytokine increase was seen as shown in the BRGSFis model. Just to conclude now, how this type of CRS study compares to what is done in PBMC reconstituted model. As you know, many people use PBMC reconstitution to study safetiness of antibodies. We decided to compare the two systems. So we took BRGSFis mice, which were reconstituted with cord blood cells, and we took mice reconstituted with human PBMCs. First thing we did was just to compare the immune population without any treatment. This is what we observed. In the PBMC reconstitution in that study, 50% of the human cells were T cells, 31% were B cells. In the cord blood cell reconstitution, it was the opposite. We had 13% of B cells, of T cells, and a majority of B cells, as reported in any HIS model. Then we look into the activation status of the T cells. This was done 2 weeks post-PBMC injection, 2 weeks only. At this time, you have no clinical sign of GVHD in PBMC reconstituted mice. Looking into this activation status, this is what we found. In CD34 positive cells, naive cells represent 30% of the T cells, whereas in PBMCs, they represent only 3%. Looking now into the effector memory cells, they represent 41% here versus 70% here. So we have much more activated cells in the PBMC model. Second, when looking into those cells, they are LAX3I, CD25I, so they are already pre-activated T cells. How does this translate in terms of cytokine profile in a CRS study? The two models were treated with OKT3, and we look into the cytokine production. The higher level of cytokine produced were in the PBMC model. Surprisingly, yes. Why is that? Very simple. When we check into gamma, your gamma baseline into the PBMC model are already super high prior to any injection. And this, in fact, reflects your T-cell activation due to the GVHD which is started. So the take-home message is, you could do a cytokine release, you'll see a nice peak of cytokine, but you are hitting T-cells which are pre-activated by that GVHD, unless you use a double knockout MHC class 1 model, which has been, for example, promoted by the JAKs. You also see inflammatory cytokines in the system, inflammatory myeloid-derived cytokines, and body weight could be seen in both, body weight loss could be seen in both systems. So clearly, we believe that a cord blood cell reconstitution will be most appropriate for CRS study than a PBMC reconstitution due to the activation status of the T cell in the PBMC reconstitution. So just to conclude here, these type of models could be quite helpful for efficacy, for investigating the PD of a compound, but also for investigating the safetiness of a compound. The immune system that I showed is functional both in terms of lymphoid and myeloid compartment. However, the interaction with trauma and epithelial will not always be functional in such a model. We are working on a novel generation of this model in which we have included human FcRn for PK study on top of efficacy study. And the model has been detailed into this poster that you could get by talking to my colleagues. However, these should not be against syngeneic model. Syngeneic models are quite helpful as well, as long as we know exactly how they have been generated and what are the limitations of these models, because there's many target humanized models which are around, where immune checkpoints were humanized, where CD3 has been humanized, where Fc gamma receptor have been humanized. They could be a very valuable tool, as shown for the Vista knock-in mice. Here, the whole crosstalk will be functional. We no longer have to focus about stroma or whatever. That works, as long as you have properly validated that your human gene function into mouse cells and that it doesn't modify interaction with other partners. This is why we decided to keep next to the HIS mice syngeneic mice which are fully human. And in order to enhance the translatability of these models, we also included into our catalog models which have a human CD3 for T-cell engagers. This model could also show cytokine release, models which have Fc gamma receptor humanized, and models which have as well humanization of key components of the PK pathways such as FcRn and HSA, and as well as models where we've humanized FcRn, TFRC for drug shuttling the BBB, and serum albumin for more translatable PK/PD. So as you can see, our motto is really to try to move toward the most translational model as possible, although it's remain an animal model, and there will still be major discrepancy when compared to human. So I'd just like to finish here by thanking our colleague at genOway who contributed to this work, and also all our partners who were involved into some of the studies that I mentioned, including our colleague from Sensei for the work on the Vista, but also CronBio for the CRS work, and all the other pharma partners such as OncoDesign and the others. So this is what I wanted to share with you. We have time for a few questions, I believe. We have 15 minutes left. And you could always stop at our booth this afternoon. And we have 2 posters this afternoon on CD28 and Fc gamma receptor humanized mice. So feel free to stop by if you have any questions. Thank you for your time, and thank you for stopping by. Any question?
Kader, thank you so much for your presentation. You mentioned that in the tumor bed, you have human as well as murine immune cells. What's the functional state of those murine immune cells? Is it dependent on what type of cancer models are being run, or is there a crosstalk? Are they functionally contributing as well?
It's a very good question. In fact, when the FLT3 knockout mice, I'm talking about syngeneic mice, okay, were generated, The only thing that was reported is a drastic reduction into FLT3, into myeloid cells. What has to be done now, it's really to know how this reduction into myeloid cells correlate to their function status-wise. Okay? We know that affecting the FLT3 ligand pathway would indeed modify some of the maturation of the mouse myeloid cells. But we don't really know which function are affected and which function are not. What we know for sure is that the neutrophil part is unaffected. So any ADCC triggered by mouse neutrophil could still contribute to the readout we're having. Okay? And that's something we want to do indeed, is to better characterize what the remaining mouse cell could do in the system and how they could contribute to the immune response, but for sure, they contribute to the immune response as well.
Hypotheses and therapeutics. Thank you very much.
Thank you, Kamal. Thank you. Any other question? So thank you all for your time. Thank you for stopping by.